We are starting with a very important topic in endocrinology, which is the pancreas. Now, pancreas, as you all know, it's a long, slender organ, most of which is located posterior to the bottom half of the stomach. And although it is primarily an exocrine gland, the exocrine portion always secrete digestive enzymes, digestive juices, which we have discussed in depth in the GIT physiology. Now, the endocrine portion, they are known as pancreatic islets of Langerhans, which secretes the hormone insulin, glucagon, somatostatin, and pancreatic polypeptide. So, if I talk about the histology of a pancreatic islets of Langerhans, so we have alpha cells, beta cells, delta cells, and the F cells. And the alpha cells produces the hormone glucagon, and they make up 20% of each islet. So, this glucagon raises the blood glucose level by stimulating the liver, which converts the glycogen into glucose. Okay, and beta cells, which produces the hormone insulin and makes up approximately 75% of each islet. And this insulin lowers the blood glucose level. The delta cell accounts for only 4 to 5% of the islet cells, and they secrete somatostatin. Now, this somatostatin is also released by the hypothalamus, and the stomach and the intestine also secreted it. Somatostatin suppresses the release of other hormones which are made in pancreas. So, and there are another type of cells, the pancreatic polypeptide cells, which is also known as F cells, and they secrete pancreatic polypeptide, and it accounts for only 1% and secretes the pancreatic polypeptide hormone. Now, this hormone is thought to play a role in appetite by increasing the appetite. So this hormone released following a meal may reduce further food consumption. So this is always released in response to fasting. So pancreatic polypeptide is released always following a meal. Then after a person has or had a meal, that after the secretion of this hormone after a meal, it may reduce the further food consumption. So if I see over here in this diagram, I can see the alpha cells, I can see the islet cells, I can see the pancreatic acini also, the exocrine portion, then I can see the delta cells, the beta cells as well. Now, if you remember, what do you mean by paracrine control? Paracrine mein what happens, this insulin activates the beta cells. Okay, so as you can see, this insulin activates the cells, and then it inhibits the, so as you can see in this diagram, the insulin activates the beta cells and inhibit the alpha cells. This red arrow is for inhibiting. So the insulin hormone activates the beta cells and inhibit the alpha cells. The hormone glucagon, as you can see, this activates the alpha cell. This one activates the alpha cell, and when also activates the beta cell as well. So here we can see two light green arrows. So this activates the beta cells also, and then it activates the alpha cell also. Now this also activates the delta cell, which is secreting the hormone somatostatin. But the somatostatin hormone inhibits the alpha cells and the beta cells. So if you remember, there is this paracrine control which coordinates the secretion of this pancreatic polypeptide. So all these are closely interrelated. That's why I told you in this diagram, nothing so much important for your exam purpose, but it's just a close relationship between all the three types of cells. Now, so if I come to insulin, the primary function of insulin is to facilitate the uptake of glucose into the body cells. And I've already mentioned that this is secreted by the beta cells. So what are the actions of the insulin on the carbohydrate metabolism? So this insulin increases the transport and the uptake of the glucose by the cells. So by increasing the permeability of the cell membrane to glucose. This is the major and the most important function of insulin, by increasing the permeability. So this also increases the number of glucose transporters. So I've already mentioned about a glucose transporter named as GLUT in the cell membrane. So all the functions of the insulin are to increase the uptake of the glucose into the body cells. So all the red blood cells as well as the cells of the brain, liver, kidneys and the lining of the small intestine, they do not have insulin receptors on their cell membrane and do not require insulin for the glucose uptake. Although the body cells do require insulin if they are to take glucose from the bloodstream, skeletal muscle cell and adipose cells are the primary targets of the insulin. Now, the presence of the food in the intestine triggers the release of the GIT hormones such as glucose-dependent insulinotropic peptide. Fine. Now, this is in turn the initial trigger for the insulin production. So, what happens whenever we ingest a food? Then, what happens? The GIT, it henceforth increases the secretion of the insulin by stimulating a glucose-dependent insulinotropic peptide. And then henceforth secretion by the beta cells of the pancreas. Once the nutrient absorption occurs, the resulting surge means always the increase, the resulting increase in the blood glucose level, it further stimulates the insulin secretion. So the second function on the carbohydrate metabolism is helpful in the storage of the glycogen. If the muscles are not exercising after a meal, then glucose is transported into the muscle cells. So, into the muscle cells and stored in the form of muscle glycogen. Now when do we require under two conditions the muscles use glucose. First, during his heavy exercise. Now this usage does not require insulin because the muscles become permeable automatically to more and more amount of glucose. So they do not require insulin that time when the muscle is doing some heavy exercises. And also few hours after the meal, blood glucose concentration increases and insulin also increases to combat the increased glucose level in the blood. So henceforth this glucose is also transported into the muscle and then it is stored into glycogen form. The third function is the insulin promotes the liver uptake, storage and the use of glucose. So whenever the food is not available, the glucose drips down and insulin also dips down and then glycogen is split into glucose. So one of the most important of all the effect is to cause most of the glucose which is absorbed after a meal to be stored in the form of a glycogen. Now, these are some major important points which you have to keep in mind. What happens? This is the mechanism by which the insulin causes the glucose uptake and the storage. So, what happens? First, the insulin inactivates the liver phosphorylase enzyme. This is the enzyme which causes the liver glycogen to split into glucose. What will happen? This will inactivate this enzyme. So, उससे क्या होगा? The glycogen का breakdown नहीं होगा. The second point, insulin causes the enhanced uptake of glucose from the blood by the liver cells and does this by increasing the activity of the enzyme glucokinase, which is one of the enzymes that causes the phosphorylation of the glucose after it diffuses into the liver cell. Go back to your biochemistry lectures and you can very well correlate with all these enzymes. So this insulin increases the activity of the enzyme glucokinase, which causes the phosphorylation of the glucose. Now the phosphorylated glucose cannot diffuse back through the cell membrane. That is why it increases the activity of this enzyme, because once the glucose is phosphorylated, it cannot diffuse back to the cell membrane. Now, insulin also increases the activity of the enzyme which promote the glycogen synthesis, especially the hormone known as glycogen synthase. So, net effect, increase glycogen in the liver. So, glucose is released from the liver between the meals. So, less blood glucose, decrease insulin secretion. The lack of insulin reverses all the effects. अब once जैसे ही इंसुलिन का इफेक्ट कम होगा, once the insulin secretion has dropped down, जितने भी effects हमने अभी पढ़े, they all be reversed. So what will happen? The lack of insulin activate the enzyme phosphorylase, which was initially inactivated. Now this causes the splitting of the glycogen into glucose phosphatase, and the enzyme glucose phosphatase, which had been initiated by insulin. Now this allows the free glucose to diffuse back into the blood, and henceforth this whole thing is managed between the insulin and the glucose concentration. So the fourth point in the carbohydrate metabolism, already we have talked about the 3 points. ठीक है, increases the transport and uptake of glucose, storage of the glycogen, and the use of the glucose by always converting this glucose into the glycogen by all these effects or these actions. The fourth point is the insulin promote the conversion of the excess glucose into fatty acid, and this will inhibit the gluconeogenesis in the liver. Now, whenever the quantity of glucose is more, then what happens? The insulin promote the conversion of all this excess glucose into fatty acids. So, henceforth, this will inhibit the gluconeogenesis. So, it increases the peripheral utilization of the glucose. So, if I have to sum up, I can say it increases the peripheral utilization of the glucose, which means all the cells are taking up the glucose. It increases the storage of the glucose by converting into glycogen. It inhibits the glycogenolysis. Glycogenolysis means glycogen, the breaking down of the glycogen, and it inhibits the gluconeogenesis. The second effect is on the fat metabolism. But you have to remember in this that the insulin activates always all these enzymes. So the name of the enzymes are not given in your book, so you just have to memorize all of them. If written in your exams, this will help you in scoring more. So the next effect in the fat metabolism is that the insulin promote the fat synthesis and the storage. So this will increase the transport of the glucose into the liver cells. The glucose is first split into the pyruvate in the glycolytic pathway, and then what happened? And then the pyruvate is converted into acetyl coenzyme A. This acetyl coenzyme A is a substrate from which the fatty acids are synthesized. So the most of the fatty acids are then synthesized within the liver itself, and they are used to form triglyceride, which is the usual form of the storage form of the fat, and henceforth they are released from the liver cells to the blood in the lipoproteins form. So this is the whole pathway. So this insulin activates the lipoprotein lipase in the adipose tissue. What will happen? Once the lipoprotein lipase is activated in the adipose tissue, this will split the triglyceride into fatty acids, and then these fatty acids are again converted into triglyceride, and then they are stored. So the stored of the fat in the adipose cells by inhibiting the action of the hormone sensitive lipase, lipoprotein lipase को inhibit कर देंगे. By inhibiting the action of the lipoprotein or the hormone sensitive lipase, which causes the breakdown of the triglyceride. So henceforth this will, अगर हमने hormone sensitive lipase को inhibit कर दिया, then what will happen? The storage of the fat in the adipose cells will happen. So this promote the fat synthesis and the storage. As I've already mentioned, this insulin activates the lipoprotein lipase in the adipose tissue. Now this will split the triglyceride into fatty acids, and then so what will happen by inhibiting the action of hormone sensitive lipase, there is no breakdown of the triglyceride. On the protein metabolism, this increases the transport of the amino acid into the cell from the blood by increasing the permeability of the cell membrane for the amino acids. It helps in increasing the protein synthesis. It prevents the protein catabolism. So the main function is the anabolic action on the protein. It prevents the conversion of the proteins into glucose. So transport of amino acids by increasing the permeability, more and more of protein synthesis by transcription and translation, and preventing the protein catabolism. Also on the growth, because insulin growth hormone, they interact synergistically, that means they interact together to promote the growth. So increases the transport of the amino acids into the cell and synthesis of the protein in the cells. So if I have to sum up, what I can say, on the carbohydrate, it facilitates the glucose entry, it stimulates glycogenesis, it inhibits the glycogenolysis and causes or inhibit the gluconeogenesis also. On the fat, it increases the transport into the adipose tissue, it promotes the triglyceride synthesis, and it decreases or inhibits the lipolysis, the lipid breakdown. In the protein, it promotes the uptake of amino acids, causes the protein synthesis, and inhibit the protein degradation. Now one more important point which you have to keep in mind in insulin is the chemical structure. So insulin receptor is a tetramer. As you can see, it's a tetramer formed by 2 alpha unit and 2 beta subunits. Fine. The alpha subunits, they protrude out of the cell and the beta subunits are inside the cell. Okay. So they are linked by disulfide bonds. As you can see, they are linked by disulfide bonds. Okay. So intracellular surfaces of the alpha subunits, they have this enzyme protein kinase or the tyrosine kinase enzyme. With the help of this enzyme, the further actions of the insulin are taking place. But just you have to keep in mind the main diagram of the insulin.